for Alzheimer's disease treatment.
Very good.
Very good results.
Good.
Okay, so as many of the presenters, I would like first of all to say thank you for the organizing of this very interesting meeting, and of course for the host of this meeting. It's a great pleasure for me to meet again old friends and colleagues here, and to see such a wonderful place like the Basque countries. So again, thank you very much. The title of my presentation is Novel Multitarget Compounds as Potential Agents for Alzheimer's Disease Treatment. During these days, we have discussed a lot about importance of multimodal regulation of brain in the stress or in disease conditions. So I will also touch this problem, but from the point of view of medicinal chemistry, because I'm mainly a medicinal chemist. Okay. So 2 words about Alzheimer's disease. Of course, you know a lot about Alzheimer's disease, and the main pathological features of Alzheimer's disease. It is beta amyloid plaques, which is formed out of the cell, and neurofilament tangles, which are formed inside the cells. We also talk a lot about it, so I will skip explanation of the slide. I would like also to stress that it is a really big social and medicinal problems now. And even in Russia, we also have a lot of people who have this problem. So it is estimated as about 1.5 million people of Russian population is sick with Alzheimer's disease or Alzheimer's disease-like dementia. So nevertheless, there are only a few compounds, only a few drugs which are now used for treatment of Alzheimer's disease. Actually, there are 4 compounds. It is donepezil, rivastigmine, galantamine, and memantine. 3 of them, donepezil, rivastigmine, and galantamine, is inhibitors of acetylcholinesterase, and memantine is the last compound which was proposed for Alzheimer's disease treatment. It is low affinity blockers of NMDA receptors. The market for these drugs is very big. It's estimated now is about $5 billion per year, and it is expected that it will be growing, maybe not too fast, but systematically, and will growing up to about $10-12 billion until 2020. Quite recently, I and my colleagues will make an analysis of compounds which now are in clinical trials. This is the data which we have published this year in Russian journals, but also we submitted our review to the Medicinal Research Reviews, and I hope it will be published also this year. So at the end of the 2015, there was about 130 compounds on different phases of clinical trials. 50 compounds on phase 1 clinical trials, 59 on phase 2 clinical trials, and about 20 on phase 3 clinical trials. It's possible quite roughly to classify these compounds on the mode of their action. So the first column, dark blue, it is anti-amyloid agents, which in any form block the process of beta amyloid formation. The second column, it is violet. It is microtubular structural target agent, which again in any formers decrease the formation of microtubular tangles. Then it is compounds which has a non-neurotrophic mode of action. Most of all, it is compounds which in any form modulate the signaling system. It is ligands, different ligands of receptors, neuronal receptors, ligands of NMDA receptors, of different forms of glutamate receptors, cholinergic receptors, GABA receptors, etc., etc., etc. So you can see that most of compounds which are on clinical trials now, it is compounds which can be called as a neuromodular signaling system. Also, there are a lot of compounds with different mechanism of action, in particular antioxidants and some other compounds. And of course, there are some compounds which mechanism of action and structure are not disclosed by the company. So what is very important, that about 30 compounds has a multi-target mechanism of action. It is proven and it is shown that this target involved in pathogenesis of Alzheimer's disease. And also about 30 compounds, these compounds which were launched earlier for treatment different other diseases, and they were repositioned for treatment of Alzheimer's disease-like disorder. So again, very few words about main hypothesis of Alzheimer's disease. So now there are at least two main hypotheses. One related to beta amyloid formation, pathological oligopeptide, which produced from the beta amyloid precursor protein, APP protein, due to processing by three at least enzymes, this beta amyloid, beta secretase and gamma secretase, and it produces its toxic action to neuronal cells in being in the form of toxic oligomers. Another point of view, it is the so-called tau hypothesis, where the main subject of Alzheimer's disease and disorder development related or attributed to tau pathology, to this formation of neurofibril tangles. At the present time, the special term was defined, so-called disease-modifying agent. It is the compound which can block development of a disorder, and if we treat the patient with such compound, the improvement of cognition and memory will still continue even after stopping the action of treating with these agents. So another slide I will not discuss in detail. I would like only to show the number of potential targets which already proven to be involved in Alzheimer's disease. I took this cartoon from the database of process integrity, and there are about more than 100 target molecular structures which have been shown and proven which participate in the development of Alzheimer's disease. So, multifunctional nature of Alzheimer's disease is, there is no doubt of multifactorial nature of Alzheimer's disease. And when we can just look on this picture, we can understand that development of compound which will interact with only one single target, it is very, very problematic that it will show the beneficial effects when the disease is so multifactorial. So one of the main trends now in the development of new compound for Alzheimer's disease treatment, it is the development and discovery of so-called multifactorial or multi-target or multi-ligand. There are a lot of different terms, but the meaning is the same. It is the approach which is based on the attempt to combine in one chemical structure different pharmacophores which interact with the target involved in the pathogenesis of Alzheimer's disease. So it really looks quite attractive, but as usually, good idea sometimes has a lot of problem in its practical realization. So there were a lot of attempts to design compounds which will unite in one structure different effective pharmacophores. A lot of attempts were involved or have been made with the Bachurin, the first compound which was proposed for Alzheimer's disease treatment. So there was an attempt to conjugate to Bachurin lipoic acid, or calcium channel blocker nimodipine. All of them, they showed quite good results in vitro and in vivo animal experiments, but none of them passed even phase 2 clinical trials. Only one more or less successful compound, it is so-called it combined two main fragments. One is inhibitor of acetylcholinesterase, rivastigmine, and another is rasogeline. It is the inhibitor of monoamine state B. So he combined these two templates in one structure, called lagostagil. But it failed in phase 3 clinical trial, but now they move this compound back on mild cognitive impairment treatment. I may say that it is also quite a general tendency, trend in development of compound. When companies see that a compound looks quite beneficial and show some beneficial effects on phase 1 or phase 2 clinical trials for Alzheimer disease, but failed on phase 3, they try to move it back on mild cognitive impairments. But it's because it is much more has much more probable to be beneficial for treatment the very early stages of Alzheimer disease than to treat the mild or moderate stages of Alzheimer disease. So now I would like to show some examples which came from our institution. So this work was completed some years ago, and the first part of this work I presented some years ago also on the ICNA meeting. So we try to construct compound which will simultaneously act on two main inotropic glutamate receptors, NMDA receptors and AMPA receptors. It is known, and also we have heard a lot about it during this conference, that glutamate receptor system play a very important role in neuronal plasticity, in particular in memory consolidation and reconsolidation process. But under overstimulation of NMDA receptor, first of all, due to different stress factors of beta amyloid, this calcium overloading of cell results in a number of toxic processes in the cell, in particular apoptosis, necrosis, etc., and the result cell death. So the idea was to construct compound which from one hand which will block NMDA receptors and therefore prevent renal cell death after this excitotoxic action of stress factors of beta amyloid. And from the other hand, it will activate AMPA receptors. We decided that just to combine NMDA receptors blockers and agonists of AMPA receptors is not too promising due to also some excitotoxicity of AMPA agonists. So we select not AMPA agonists, but AMPA positive modulators. So we combine NMDA receptor blocker and positive modulator of AMPA receptors. Positive modulator is a compound which does not activate itself AMPA receptor, but it increases the response of AMPA receptors to endogenous glutamate. So to skip chemical part of this work, which was completed 2 years ago, we construct compound. Lead compound was NT1505. We shown first of all that by molecular docking that it very good fit the intrachannel site of NMDA receptors and also allosteric site of AMPA receptors. It showed quite low toxicity and quite high efficacy in different animal model of dementia. In particular, we use in this case Morris water maze test and use animals which were treated before with neurotoxin AF648, which selectively destroy cholinergic neurons in basal nucleus minor. So we showed that this compound really can be discussed as potential drug candidate. Now we prepared all documents for clinical trials, and we're waiting when our ministry give the permission to start these clinical trials. Another compound, another example, is also the ligand of glutamate receptors, but another approach. It is known that AMPA receptor itself, it has different subunits, and each subunit has a symmetrical site where possible to modulate, which permit to modulate allosterically the activity of AMPA receptors. And there are a lot of compounds which have been also discussed as potential cognition enhancers. It's called AMPA kinins, which interact with each side of this AMPA receptor subunits. So we designed the compound which simultaneously interact with the both sides of AMPA receptor. So we took the structure of one ampakine and linked it to another structure of the same ampakine with this proper spacer. It is diazobit-cyclonone. Finally, we also select a lot of compounds and select the lead compound, which show the AMPA potentiating activity in very low concentration. It's about 1 nanomoles or even picomoles. So it's the record among the different ampakines. What was very interesting and unexpected, that this compound shows also quite unusual property to restore the lost or weak memory. I will try to explain this experiment very briefly. So there was taken chicken, and chicken, of course, they from the very beginning they know how to dig the seeds. So we put one part of the seeds in the bitter solution, and after some training, chicken could select good seeds and bad seeds. So they selected according to their color. So the bitter treated seeds were colored by another color. It's green, red, and so, and after 24 hours, chicken still remember that there are good seeds and bad seeds, and they select only good seeds. But if we wait one more day, they forget about the difference in seeds. But if this chicken were treated by our compounds 5 minutes before their training procedure, or even, it is especially important, 4 hours after training, they still keep in mind these differences between two seeds. So it looks like that they forget about it, but when they took this compound, they remind the differences between good and bad food. It's an unusual property, and now we are just thinking about how to use it in practice. It could be very practically used by students. Just took this pill before the exam and remind everything that you have read before. So, but of course, it is not so easy to move such kind of compound to the clinical trials. Another group of compounds was constructed on the ground of preliminary results which were received by us and our colleagues from Japan. So they showed that dimibond compound which were invented in our laboratory, it's gamma-carbolin and methylene blue. Each of these compounds and being introduced in mixture, they significantly decrease the aggregation of different pathological proteins in the brain. In particular, here they show the results with TDP-43 protein, which model ALS disorder. So it gives us ground to construct compounds which combine in one structure templates of dimibond, this gamma-carbolin, and methylene blue. So we chemically synthesize different chimeras which contain both these templates, and test how these compounds interact with different molecular targets, which were specific to both these primary pharmacophores. So we have shown that in contrast to original compounds, these hemers compounds, dimers compounds, they showed very selective inhibitor activity towards butyrylcholinesterase. We also made three-dimensional docking of interacting these compounds with active center of butyrylcholinesterase, and shows that the major interaction was piece taken between indole ring of butyrylcholinesterase and a phenothiazine fragment of this compound. Also, we test how this group of compounds interact with glutamatergic receptors, in particular NMDA and epinephrine receptors. So we showed this compound also possessed quite strong activity against epinephrine receptors and quite moderate activity against intrachannel side of NMDA receptors. It is interesting is that none of the preliminary structure, nor dimethyl nor methylene blue, did not show this combination of properties against different receptors. So finally we select two hit compounds, and now we're completing preclinical trials of this compound in different animal models. So if we will be lucky, we will also be able to move to other clinical trials. And the very last example. Also the ground for these results were taken from the preliminary experiments which were performed by a group from the United States. It's the laboratory of Stephen McKnight and Andrew Pieper. So they synthesized and showed that none of carbazole derivatives is on the upper part of the slide. They possess so-called pro-neurogenic activity. They induce neurogenesis in the cell, and also they show quite strong neuroprotective activity. Due to the quite close structural similarity between these compounds and our compound dimebone, they also test for any case how dimebone interact in this system. And also they showed that dimebone as well as their compounds show quite a strong pro-neuroprotective activity. So we from our side decided to combine both these templates, carbazoles and dimebones. So dimebone and carbazoles. So we synthesized compound which contain both these pharmacophores and showed that it also has the specific properties of both these compounds, but what was again unexpected and unpredicted. It shows very new ability to potentiate the process of polymerization of tubulin into microtubules. And what is especially important, these microtubules, which was formed due to this induction of polymerization of tubulin, were formed in a very correct form. So we checked different compounds, and a lot of compounds also can influence the polymerization of tubulin. But in most cases, this polymerization results in very random structure of the microtubules. In this case, we received the microtubules which are very typical to the control condition, to the condition of normal breeding. So it looks very attractive, and finally we performed some behavioral experiments. In this slide I show the very last data which we received again in modest water maze test. We took a heat compound DF302 and showed that in modest water maze test it showed quite strong cognition enhancing property. So what is the conclusion? Such a specific conclusion which could be done from these results. The design of multi-pharmacophore agents interacting with the key stages of neurodegenerative process provide diversity of potential neuroactive compounds with variable spectrum of activity. What is interesting and what should be taken into consideration, in some cases, the pharmacological profile of conjugates of pharmacophore fragments could be notably different from the profile of basic structure. And in the specific case of our results, we show that new conjugates of gamma-carbonins with phenothiazines or carbazoles give ground to a development on this base new potential agent for complex protection of cell against neurodegeneration typical to Alzheimer's disease. And the very last slide, I just tried to formulate, at least for myself, what is good and what is bad in multi-target strategy. So of course in picture everything is understandable. It's better to close the whole roof than to close each spot on the roof when it is broken. So, but any of the strategies has its positive and negative sides. In monotarget strategy, what is good? It is a relatively simple design of structural optimization. It is very easy. Convenient models for in vivo modeling, in vivo and in vitro modeling. Exact efficient doses. What is contra? Non-adequate to pathogenesis. Again, I would like to stress that any neurodegenerative disorder, in particular Alzheimer's disease, it is multifunctional disorder which involve a lot of molecular target in the pathogenesis process. More side effect if we will use combination therapy. And incompatibility of the agents. So it is the contra to the multitraget drug. What about multitraget stage? What is contra? Relatively complexity in design and structural optimization. It is really quite difficult for chemists to optimize structure when this structure binds a lot of different pharmacophores. Complex in vivo model. Of course, in this case, we can use only cell or animal models, not in vivo model. And the most important and most problematic point is the different efficient doses at different times. It is the main problem of multi-target agent, because each agent, it has its own effective doses, and when we combine in one structure two pharmacophores, it means that we need that these compounds, so these two pharmacophores or three pharmacophores, should act on the same dose. So it means that when we start to construct these compounds, we should select the pharmacophores which has very close efficient dose. Or in any case, we will not solve this problem. And what is positive, it is told from the very beginning about it, it's more adequate to pathogenesis, less side effect, and expanding of pharmacological profiling. So each of medicine chemists should decide what is better for him in each case. Thank you very much.
Thank you, Sergey. Do we have some questions for you? And one thing I noticed, Sergey, is that years ago when this conference was sort of mainly focused on trying to treat stroke, that this same sort of maturation occurred. We went from single compounds to multiple compounds because of the complexity of the aftermath of stroke. And here you're certainly seeing the same kind of tendencies where you're talking about building compounds that have multiple effects that are desired to treat the overall syndrome. So that's very interesting to see that. So any other comments from the audience?
Well,
Yeah,
it
Russ.
seems like a big problem. A lot of pharmacological agents that you take orally or inject, it goes everywhere. And so you're basically hoping the effects are greater than the side effects. It seems like as you get multi-targeted, that's even more of a problem. Because, I mean, so much of this is depending upon time and space and the nervous system or its efficacy. I think some people are working on techniques to more localize the distribution of pharmacological agents within certain areas of the brain.
Well, of course, I agree that in any case, when we when we synthesize a new compound, in any case, it should have a by-side effect. Of course, this doesn't mean that this by-side effect of each pharmacophore will be multiplied. It's not the simple sum of this by-side effect. They could change. But in any case, we could predict or at least foresee what kind of by-side effect this multi-ligand compound should possess. So it's, from my point of view, it's not the biggest problem. The biggest problem is to combine proper pharmacophores, which has the same or very close efficient dose. Because if they will be too different in efficient dose, they will not act as a single compound.
Yeah, so I still see, say in brain tumor work, getting chemotherapeutic agents to the area of interest in high doses, so that's nanoparticles and various mechanisms for targeting. That doesn't seem to be happening nearly as much in Alzheimer's disease. Targeting the areas that should be targeted with these. So maybe one drug would be sufficient if it could be in the right dose, the right places.
Maybe, but you see that the very last compound which was launched on the market was in 2003, and a lot of money was spent on developing new compounds. Each year there was about 100 clinical trials, and none of them from 2003 was successful. So, and many of these compounds, they explore the idea of monotouched action. So, of course, there is no guarantee, of course.
Yeah.
No guarantee, a big risk and a lot of potential problems. But it is one of alternative. in developing something new, which could be promising.
Yes, please.